Hi, my name is Corey Callaghan, and I wanted to talk to you about a recent project that we have published titled Capitalizing on Opportunistic Citizen Science Data to Monitor Urban Biodiversity, a multi-taxa framework. We use data from the City Nature Challenge in the Boston area to understand how species are responding to urbanization. To do this, we took all the data submitted by iNaturalist users that were research grade and available in GBIF, and we overlaid those observations with a measure of urbanization. We used VIIRS nighttime lights to do this. So in the middle of downtown Boston, you have the most highest values, whereas in the very forested natural areas, you have much lower values. And so each species was, based on their observations, was left with this distribution in response to nighttime lights, measured as average radians. So here are 8 example species showing their distribution in response to urbanization. Some species generally don't use urban areas, like Canada mayflower and spring peeper, whereas other species actually fare pretty well, and a lot of their observations come from urban areas, like eastern gray squirrel and Japanese knotweed. We did this for all the species that we had enough data for, and we ended up with more than 1,000 species that we were able to quantify this urban tolerance score. And here's a distribution of all those urban tolerance scores on a log scale. As you can see, you have a range of responses by different species, such as the eastern gray squirrel, which is way out here and really one of the more urban species, and the spring peeper again, which is much less urban, but a lot of species that are in between. And what that allows us to do is understand how different areas are actually influencing this urbanness community. So we were able to take the mean of all the observations in a given area and take the mean urbanness value to get an understanding of how urban that community is. So here on the lower end here, we have this less urban community, which is a lot of species that occur only in natural areas. And then it ranges all the way to this more urban community, which is dominated by species that are exotic species that are really urban. And as you can see, we looked at the relationship with this town biodiversity urbanness index with EVI, which is enhanced vegetation index, impervious surface, and the percent tree cover in an area. And unsurprisingly, as you have more EVI and more tree cover, you get more non-urban species. And conversely, as you have increase in impervious surface, you get more urban species. This is really important and helps us understand how biodiversity is responding to urbanization at both species and community levels. Citizen science data contributed by participants like yourselves through iNaturalist can definitely assist in biodiversity monitoring within urban areas. These iNaturalist observations are really valuable for researchers like me and who are urban ecologists, and also for urban planners. And this is really important information to continue contributing to citizen science platforms such as iNaturalist. Importantly, sampling in both urban and non-urban areas, as well as common and uncommon species, helps us better understand how biodiversity is responding to urbanization.